Today we're going to be talking about thyroid peroxidase, also known as TPO, and the role it plays in the production of thyroid hormone. Thyroid hormone production starts in the hypothalamus, then to the pituitary gland, and then into the thyroid gland. In the hypothalamus, there are a pair of ventricular neurons that secrete hormone called thyrotropin-releasing hormone, TRH. TRH enters the anterior pituitary gland and stimulates thyrotrope cells, which secrete another hormone called thyroid-stimulating hormone, also known as TSH. TSH is released into the bloodstream and travels to the thyroid gland. Let's take a closer look at the thyroid gland. Represented in red are blood vessels. Then we have follicular cells, which make up a follicle, and inside these follicles is the colloid. TSH binds to follicular cells, which this initiates the thyroid hormone production. Let's take a closer look. TSH is in the blood, binds to a TSH receptor. This allows thyroglobulin to enter the colloid. Thyroglobulin has these tyrosine amino acid residues, which are important for later. Iodide is in the blood, and it needs to get into the follicular cell, but it needs to travel against the concentration gradient, so it uses a sodium iodide symporter. Iodide then gets into the colloid using a pendrin transporter, and then it is oxidized by TPO into iodine. Then is iodination, where TPO attaches iodine to the tyrosine residues on thyroglobulin. These residues are then coupled to give T3 or T4, which are thyroid hormones. These thyroid hormones can then travel into the blood and be distributed. So, TPO does oxidation, iodination, and coupling. We're going to be focusing on oxidation in this video. Let's take a closer look at the TPO enzyme. Here we have an intracellular domain, a transmembrane domain, an epidermal growth factor domain, a complement control protein, catalytically active domain, N-terminal propeptide domain, and the heme-containing domain, which is important for oxidation, as hydrogen peroxide binds there, and then iodine comes and is stabilized by either hydrogen bonding or van der Waals forces in the active site. This is the heme. In our mechanism, it's going to be represented as Fe3+, just to make it simpler, because this is what the heme actually looks like. It's an iron surrounded by 4 chiral rings. Alright, now that we know the basics, let's get into oxidation. Yo, TPO. Let's kick it. Alright, stop. Collaborate and listen. TPO's back with an oxidation mission. Heme groups got iron in tightly, needing hydrogen peroxide daily and nightly. Iodide rolls in through sodium symport. Hydrogen peroxide comes in tight, kicking off of water, but that's alright. Binds to the heme, now we're ready to ignite. Feral species, that's the attractions. Electrons flow, this is a daily reaction. Iodide's filling up that negative space, moving in on feral whom it's gonna replace, resulting in iodine or hypodious acid. Either way, the thyroid peroxidase don't play. Feral regenerates again for iodination, to sum it up with a coupling reaction. Now we know about oxidation, let's kick it and get into iodination. Iodine is our reactive species. Feel that energy, ready to attack, attracted to tyrosine on thyroglobulin. It goes crazy when it sees a tyrosine. Stacking up, mono or di, MIT, DIT in the mix. Couple them together, it's a hormone fix. Did it stop? No, it just keeps going. Into the bloodstream it flows. Into the muscles and organs it goes, won't have no energy lows. TPO, oxidize, iodide, let it go, turn into iodine, watch it flow. TPO, catalyze, now you know, thyroid hormones rising, watch them grow. So remember this cycle, it's a crucial path. With TPO in action, feel the metabolic math. From iodide to iodine, that's the game. TPO, the enzyme, now you know its name.